start to break. And so the molecule destabilizes, and then the molecule stretches, and it changes the active site. Right? pH is the same thing. Those extra hydrogens, or those extra hydroxides, start attaching to the enzyme and creating new bonds which screw out the shape. So if you increase the acidity or the alkalinity, you're going to screw out the enzyme activity. And you can throw in some graphs there to get some extra points, because that's the part, also part C. Same, oh, by the way. So part B, you have to talk about pH and temperature as the two factors that can change the shape of the enzyme and therefore screw it up. Also, if you can get more points here by throwing here non-competitive inhibitors. Remember that the enzyme has an active site, but there's another way to change its shape. Some enzymes have a secondary hole where something connects to that and changes the entire shape of the enzyme. That is a non-competitive inhibitor, remember? So it's a third way to change the enzyme's function by doing what? Connecting something else to the enzyme that makes it switch. Okay? So that's part B. That's the shape-function link for part B. Okay? Part C will ask you about the difference between competitive and versus non-competitive inhibition. So competitive versus non-competitive. This is very easy. Both things stop the enzyme from working. The competitive inhibition basically works like this. You have the substrate that's supposed to fit that hole. But then you have something else that has the same shape as the substrate. That goes in the hole, which blocks the substrate from doing what it wants to do. That's competing with the substrate for the active site. Right? Non-competitive inhibition is when you change this active site by connecting something somewhere else on the enzyme. You got that? Make sure you know the difference between the two of them for the test. And then part D, about the enzymes, asks you to pick up to 3 graphs of enzymes reactivity. Okay? For example, so part D is about graphs. Let's look at the pH graph. Remember that we did this? So I don't know if you did it in all your classes, but check it out. Part D of this question is graphs. How does pH affect enzyme activity? Oh, who knows what the graph looks like? Come on, Mary. Alright? Somebody else come to the temperature graph. Who knows how the temperature affects enzyme activity? Not straight. Do it lower, do it lower. Okay. Do the whole graph lower. Does anyone know how temperature affects enzyme activity? If it's too cold, is it going to help the enzyme? But as it warms up, does it start helping the enzyme? Yes. So it will be like this. But if it warms up too much, what happens to the enzyme? Denatures, right? So then, boom, goes down. Right? Now, what about the pH one? No. The pH, too low is bad, too high is bad, the middle is better. No, it doesn't change suddenly for pH. Yes. The pH graph looks like that. So if I increase the pH, it's too bad. If I decrease the pH, it's too bad. Optimum pH will be somewhere here. Now, while we're in here, there's another question I remember now that will show you this. pH of 3, pH of 7, enzyme A, enzyme B. Which of the two enzymes is an enzyme that works in the stomach? Which of those two enzymes is the stomach enzyme? It's enzyme A. Because your stomach has to be acidic, so the enzyme has to be able to do what? Withstand the acidity of your stomach. If it's going to work, it has to work at 3, which is your stomach acid. Right? What about which of those enzymes is the best one for the blood? What? B. Here's another thing like that. Do a temperature. This is 30 degrees. Oh, sorry. 37 degrees. This is 100 degrees. Okay? Which of these two enzymes do you find the bacteria that lives by a volcano? B. Because it's an enzyme designed to work at 100 degrees. Right? It's not a hard question. But just think about it. Okay? Don't get this screwed up because it's not supposed to be a hard question if you think about it. Alright? So, yeah, it's a graph that looks just like this. It will ask questions like that. Which of these two enzymes are most likely to be found in a bacteria that lives in a thermal vent? Obviously, the bacteria that lives in the heat, right? Which of those enzymes is better for the human body? The temperature is 37 degrees Celsius. Enzyme A. You follow? What happens if you warm up enzyme A too much? It gets Denature stops working. Alright? Now, we did a temperature and pH, but there's more. You can also do enzyme concentration and enzyme activity. If I put a lot more enzyme, it will speed up the reaction. But if I put too much enzyme, that's not going to make too much difference because I have too many workers and not enough substrate. So that means it looks like this. Right? We talked about that. The same thing is true for substrate concentration and enzyme activity. So these are the 4 graphs that you can use. Substrate concentration and enzyme activity. And you get a point for showing me the graph. Say for example, you draw the temperature graph like this, and you'll get a point. You draw the pH graph like this, and you get a point. You draw enzyme, you get a point. But if you explain the graph, you get more points. For example, you can say, this is how temperature affects the pH. If it's too cold, the enzyme has to work too slow, so it doesn't help. As you warm up, it starts helping the enzyme. But if you warm it up too much, you denature it, and you screw up the enzyme altogether. If you lower the pH too much, it changes the shape of the enzyme. If you raise the pH too much, it changes the shape of the enzyme. But if you keep the pH at optimum, the enzyme works best. Yes? Naya? No. Oh, yes, he is. He's taking a test next door. Right now? He's right here. Alright. Everybody clear on enzymes? Okay. Alright? I believe these are all the questions that I have on enzymes, so hopefully you can do a good job there. Now, water. There is another essay on water. So, there are basically almost no questions on water. The only question I remember seeing on water talks about the polarity of water. What area of water is more negative? The oxygen side. Which area is more positive? Which makes the water what type of molecule? Polar. Right? What kind of bond exists between water molecules? Between water molecules. Hydrogen. So understand, inside it's a polar covalent bond. But between the molecules it's a hydrogen bond. Okay? So the essay on water is very simple. It has 2 parts. Part A, you have to pick 3 properties of water and discuss how those properties, so you have to do this, you have to list the property, you have to explain the property, you have to give examples of the property, and then you have to tell me how it helps life. And you have to do this with 3 properties. So let's do a couple, so I can help you out here. Alright? For example, give me the property of water. One of the 6 amazing properties of water. Yes, here. What is that called? Cohesion. Alright, so I write down cohesion. Explain. Because water is polar, right? And it has hydrogen bonds, the water molecules stick together. So I named it, I explained it, which by saying molecules stick together. Now we're going to give an example of that. Surface tension. Right? Remember talking about this? Yeah. Another example. Clouds. Right? Isn't clouds water stick together? Another example. Raindrops. Raindrops falling on my head. Another example. We did this with the little paper towel. What's it called? Capillary action. Okay. How does it help life? Give me examples. How does surface tension help life? Sorry? Sorry? Cleaning. Sure. Cleaning. Also, it keeps the shape of the cell, by pushing the oils away. What else? Yes? What do you mean crops? Sure. If it wasn't for this, you wouldn't have rain for the crops. How else? Come on. What we did in class? Yes. Capillary action. So transpiration. Transpiration. So this is how the trees move water. Right? Okay. I'm not going to write the other ones, but I'll tell you the other ones. Let's do it together. Give me another property of water. Adhesion. Which is water sticking to other stuff. Examples. Water droplets sticking to the leaves, to the plant. Another example. A water droplet sticking to the glass in tube. Another example. Capillary action. How does it help life? Plants purposely leave water on their surface to make a little